Good morning. So I said to a couple of you that I would create a little video on translocation. It seems to be something that some people have been struggling with. So this shouldn't be more than 10 minutes just to make sure that everybody's got it. So the first thing to say about translocation is there's really two things you need to know. And one is the structure of the phloem, and the other is the mechanism of translocation, the mass flow hypothesis. So inside of our stem, we have two tubes. I hope everyone remembers that. The one on the right, or is it your left? The one with the green arrows on it on your screen is the xylem. And the xylem is going to be carrying the water around. Now, we're not going to talk about the xylem in a huge amount of detail today. It's going to be relevant for the translocation, but we just need to know it's there. The one without the green arrows in it is the phloem. The phloem is here. Now, you will need to know a lot about the structure of the phloem. The phloem is made of two different kinds of cells. You have the completely empty rectangles, which are called sieve tube elements, and that's written on step 1 there. Sieve tube elements. Now, sieve tube elements are just empty cells with no organelles in them whatsoever, standing on top of each other in a tube, and they have little holes in their cell membranes, the cell walls, to allow water and sugars to get through. Now, next to those, the cells with the nucleus in them are the companion cells. Now, companion cells are there because the sieve tube elements need to be kept alive. So they're not dead like the cells in the xylem, they're actually alive, but they can't create any of their own ATP or proteins or any of that, and so the companion cells over here are going to be creating twice as much ATP and proteins and all the stuff you need, which then gets pumped, half of it gets pumped into the sieve tube elements to keep them alive. So the companion cells are effectively keeping the sieve tube elements alive. Now, so these are the two tubes in the stem. The water goes in the xylem, sugars dissolved go in the phloem. And sticking out of the side of the stem will be leaves. Now obviously there's a huge amount of leaf structure we need to know, but that's not for today. So we're just going to, for now, imagine the leaves are here. Now, the point of translocation is to move sugars around the plant. We don't move glucose around the plant, we move sucrose around the plant. So the first thing to do is to turn that glucose into sucrose by binding it to a fructose molecule. So we've now got some sucrose. Now, that's not usually involved in the mark scheme for the questions, but just so we know why we're talking about sucrose. Sucrose is just a type of sugar made of glucose and fructose. It's made after photosynthesis in the leaf, and that's how we transport the sugars around the plant. So the sucrose has to be moved into the sieve tube elements, and that happens by co-transport. Okay, co-transport. Now the sugars are then going to be moved into the sieve tube element, so we now have a load of sugars in the sieve tube element near the leaf. So the ones nearest the leaf are going to be full up with sugars. Now, the point of that is it's going to drop the water potential dramatically. So if this sieve tube element here, right near the leaf, is full of sugars, it will have a very, very low water potential, a low water concentration. It will be relatively hypertonic to the surrounding environment. The xylem here is full of water, pretty much solely water, very high concentration of water. It's got a high water potential. So if you have a high water potential in the xylem and a low water potential in the phloem, then water will move from the xylem into the phloem. And that's stage 2 here. So water will move from the xylem into the phloem. And that will happen pretty much continually near the leaves. Nearly all the water, is going to move out of the xylem into the phloem, and the phloem is going to bulge out. It's going to get very large. And basically, the hydrostatic pressure is going to increase dramatically. You're packing a large amount of water into a very small space. And that can't last forever. What happens eventually is that the water, which gathers here, moves up and down the plant from high to low hydrostatic pressure. Picture stomping on a water balloon. The water goes everywhere. The water in here, which is packed into this area near the leaf, shoots up and down the plant to the roots and the shoots. And because the sucrose is dissolved in that water, the water then carries the sucrose very fast from where the leaves are to where the fruit roots and shoots are. It is then actively transported into cells in the roots and flowers, and it's going to be used for respiration, for growth, for creation of cellulose. This kind of idea for storage. Fine. So 4 stages again. You have to co-transport the sucrose into the sieve tube elements. Water then moves from the xylem into the phloem from high to low water potential, increasing the hydrostatic pressure so that the water moves up and down the plant, and then the sucrose is then actively transported into cells in the roots and flowers. Fine. Let's look at a couple of questions on this. So firstly, you've got some really nice, quite straightforward questions. If you'd like to pause me and have a go at these questions, it shouldn't take very long. So answers. Translocation is the movement of dissolved sugars. You can also move other things in it, but sugars are the main things. Then you've got explain why translocation is important. Sugars made in the leaves, they need to be moved to other parts of the plant for respiration, growth, storage. Where does active transport happen? Where does the energy get released? I think everyone could write the mitochondria. And explain why active transport is necessary in root cells. The movements of minerals and ions against their concentration gradient. And that's not really a translocation question, it's more of an active transport question. But the roots are one of the key areas we know do active transport, because they have to move very low concentrations of nutrients out of the soil and into their root hair cells. So that's just a nice short question. If you're looking for a kind of a longer, more story-style question, you're looking at this one here. Describe the mass flow hypothesis for the mechanism of translocation in plants. So that's what we've just talked about. Basically, it's asking you to regurgitate those 4 points onto a page. So can you have a go at that again? Just pause me. Right. So, answers. You're looking at, see if it works. There we go. So, in the leaf, sugars are actively transported into the phloem. Now, actively transported, you can have the word co-transport there, it's more correct anyway. It's worth noting, incidentally, in source, sometimes you'll see sink and source written down. Sink just means an area which generally needs more glucose than it creates, i.e. it's doing more respiration than photosynthesis. And source is an area that is the opposite. It does more photosynthesis than respiration, and so releases more sugar than it uses. So the source is where the sugar is coming from, where the glucose is coming from. The sink is where it's going to. Bicompanion cells is an interesting point. So don't forget the companion cells are the ones doing all the active movement, because they're the ones that are creating all the ATP. Lowers water potential of sieve cell tube and water enters by osmosis, increasing in the hydrostatic pressure, causing mass movement, and then the sugars are going to be used or converted in the root. Okay. Hopefully that's nice and straightforward. If you're still not feeling like you're not understanding that, please let me know, but hopefully that's made it easier for some people. Well, have a very good day, and I will look forward to seeing you whenever I get to see you.